Oscar. I'm a Bioassay Associate Scientist at Cellomatics. My role entails working on client projects, supporting the senior scientific team and doing some internal R&D work. My undergraduate degree is in pharmacology from the University of Nottingham. I graduated in August 2023. I work a lot with in vitro chemotaxis assays, so these are where you measure the migration of immune cells along a chemotactic gradient to sites of interest or sites of inflammation and infection. Specific immune cells such as monocytes or neutrophils are directed to follow these chemotactic gradients to sites of interest where they can either fight the infection, increase inflammation through the release of more cytokines, or increase the number of cells that migrate to the site via increased chemokine release. Chemotaxis allows cells to get to sites of infection and inflammation to combat it and restore the body to a healthy state. Different chemokines are different signals essentially, so they're very specific. So they will attract the certain cell types that have the receptors that correspond to those chemokines. This system is very complex and specific in order to best work to fight off infections. Of course, it can go wrong or it might not work. Overactive chemotaxis, which is linked in diseases such as arthritis, can cause for increased inflammation, which can lead to long-term cellular damage if left untreated. Immune cells which fail to recognize self can therefore release more chemokines and attract other cells, which in turn can release or can cause even more inflammation, potential tissue damage, as well as pain and associated symptoms. Cancer cells can leverage the mechanisms that immune cells use to migrate, which can lead them to spread around the body, and this is a potential avenue for future therapeutic treatment. And this is why producing a reproducible chemotaxis model is important. Cellomatics models in vitro chemotaxis through the use of a transwell-based system. So we load the cells into the apical chamber, and then in the basal chamber you load your chemoattractant, thus creating the density gradient of high in the basal to low in the apical that the cells can migrate downwards towards. Chemotaxis can be modulated through the use of cofactors or antagonists for the receptors that the chemokines bind to. This can be measured against the basal response or against the chemoattractant only. This allows for an accurate model that can be easily replicated and reproduced. Chemotaxis can be measured and quantified, so the number of cells can be measured through the use of commercially available kits such as CellTiter, which quantifies the amount of ATP present in live cells, thus only giving you the living cell number that has migrated. Additionally, we can use other methods such as flow cytometry to quantify the number of cells, as well as looking at specific populations within a mixed PBMC population, so specific immune cell types. We can also use fluorescence-based imaging using our Julie Stage or Pico platforms in order to visualize the cells. Cellomatics has the ability to model chemotaxis with a variety of different cell types. So we have the ability to recruit healthy donors in-house and collect blood on site, and from there we can isolate different immune cell subsets such as neutrophils, monocytes, mixed PBMCs, eosinophils, and use them in not just our chemotaxis assay but a variety of others. Additionally, we have experience using immortalized cell lines in order to model chemotaxis. So, for example, THP1 monocytes are a very popular model. Additionally, we can use differentiated HL60 cells into a variety of different cell types, such as neutrophils, as well as modeling with EOL-1 cells for eosinophils. If this sort of work resonates with your project and your project's needs, then please don't hesitate to get in contact.